or I will also aim to finish in time for questions at the end as well.
Thank you.
Okay, so in the time that we have, what I thought I would do would be to give just an overview of how we approach the problem of predicting crystal structures. A little bit about motivation, although we're going to hear a lot more about this from Anjali's just after my talk. I'll spend probably most of my time speaking about methods, and I thought the bits of the methods for structure prediction that would be most useful to speak about would be how we generate crystal structures, sort of trial structures that may correspond to what we observe when we make and crystallize a molecule, and a little bit about comparing crystal structures, because I know we have some people in the meeting who are interested in this. I'm also going to talk a bit about energies, because this guides us towards looking at where on these landscapes of predicted structures we end up observing the ones that we observe in the lab. And if I have time, then I'll speak just very briefly about dynamics in crystals. So a bit about motivation first, and this will be brief, but I thought we needed a little bit at the beginning. What we use these methods for a lot now, and the collaboration specifically with Liverpool, is to try to help in the design of functional materials. So here's an example. It's quite a nice one for a talk because it's quite visual. I have this molecule here. It's a nice chromophore in that it has electronic energy levels, which mean that it will absorb and emit light in the sort of UV visible and visible spectrum. And in this case, it absorbs in the UV ultraviolet range and emits via fluorescence in the visible range. And these are two crystals of this molecule. There are two different structures of it. And what you can see is that fluorescence, the emission, comes at different wavelengths. So you get a different colored material. Now, fluorescence might be important for building sort of optical materials, and you might want to control or design a crystal which has fluorescence at a specific wavelength. So what you would need there is you need to understand the molecular properties, because that fluorescence occurs because of the electronic structure of the molecule, but we also need to understand the solid state arrangement of those molecules in the crystal. So that yellow fluorescing crystal that we have here has molecules arranged in this way, and this blue one has the molecules arranged in a slightly different arrangement. And what we've actually done here is we've cheated. We've introduced the second molecule, which is this small one here, if you can see my pointer. That doesn't take part in the electronic transitions, so it's not affecting directly the spectrum that we're seeing. But what it is doing is it is changing the way that these molecules are arranged. Okay? So if we want to be able to design a material like this with a specific wavelength of fluorescence, you can see that we need to be able to understand how the molecules are going to pack, because it's going to change that property. Okay. Another nice example, and this also introduces another important concept, which is polymorphism, which is where one molecule, in this case, this molecule, which is nicknamed Roy in the top right corner, can crystallize into different pure packings. So there's no second molecule introduced in any of these. But what I'm showing on the slide here are 10 different crystal structures that you get when you crystallize this molecule in different ways. They're all chemically pure. They're all the same molecule. But what you can see are different properties. Color, again, and that's where the nickname Roy comes from, because we have red and we have orange and we have yellow polymorphs. So these different crystal structures are called polymorphs. More importantly, maybe if this was a drug or a material you wanted to use, you can see melting points vary by about 20 degrees across these. Okay. When I was an undergraduate and I learned about crystallization, I learned that you could characterize what molecule you had by looking up the melting point. This is all the same molecule, and melting points are all over the place. And that's telling us about different energies of those crystals, okay? But they're all chemically pure. You can also see that we have different external habits of the crystals, needles here, plates, we have blocky crystals. That would affect things like filtering if you crystallize this from a solvent and you wanted to then dry it out. Okay, so important differences between these. Okay, and we need to understand where this might occur. So crystal structure prediction then are computational methods we're developing to try to predict what structures we'll get just from a chemical diagram like what we have here in the top. via computational methods, and we want to predict the crystal structure or structures, and that's why polymorphism is important, that we might get from this molecule. So what do we hope to get out of these methods? We hope to get a complete as possible set of the crystal structures that are possible for that molecule. Okay? Not just the one crystal structure, as we show in the diagram, but because polymorphism is possible, all possible crystal structures. We want those structures to be geometrically accurate enough, because they are models, they come out of minimizing on some energy surface. Okay? But we want that energy surface to be accurate enough and reflect reality well enough that the structures we get out are close to what we'd get if we determined that structure experimentally. So close enough would be, say, close enough for structure determination. So we had some experimental data we could try to match up with for property prediction, like that fluorescence wavelength I showed a couple slides ago. Typically, we're talking about we need things in terms of atomic positions. If we took, say, a cluster or cut out a sphere of, say, 25, 30 molecules in a crystal, we want predicted structures to match experiment to within better than, say, 0.3 or 0.4 angstrom in terms of mean positions of those atoms. And that can be quite challenging, actually. We also, out of this, we don't just need structures, but we need relative stabilities, because a big assumption in this is that when we generate structures and assess them, we're going to be looking for the lowest energy possible structures for that given molecule. The big assumption is that the lowest energy structure, the global energy minimum, is the most likely crystal structure we'll see in the lab. But again, going back to polymorphism, we know that some higher energy structures may be possible under different crystallization conditions. So the energies we're typically talking about are static energies, looking at the crystal structure as if these molecules are just sitting in place, and we're calculating what is essentially equivalent to an internal energy of that crystal. But we move towards, and this is where I might get time to talk about it at the end of today, it's free energies, and that's where we need to start thinking about the way the molecules are also moving around in the crystal. Then we can look at effects of pressure and temperature on those relative energies of predicted structures. Okay, so how do we go about this? We start with a chemical diagram. We want to be able to start with only this, no experimental information. So what this is telling us is what atoms we have in our molecule and how they're bonded chemically. Okay. Our first step then is to turn that into a three-dimensional picture of our molecule. So we use electronic structure methods, quantum chemistry methods, where we calculate molecular orbitals, we fill those up with electrons, and then we optimize that three-dimensional structure where the atoms are such as to minimize the total energy of that molecule. That gives us what is at least the geometry of that molecule as an isolated entity. And we take that as a building block. And then what we do is we start to perform a structure search. And this typically involves generating trial structures on the order of tens of thousands up to millions of possible structures at the start. And we'll get to a bit more detail about this. What we use currently, mainly in our lab, are low discrepancy sequences to give a uniform coverage of all of the structural degrees of freedom that we have in a crystalline system. And we'll talk about those in a minute. Okay, so we're generating lots and lots of structures. We then need to assess these very quickly. So we screen them. Some of these from sort of a quasi-random type search will be physically unrealistic. So we may want to either reject them or adjust them. We'll then perform a local optimization, usually on just that static energy surface. So we're looking for the nearest local minimum on the energy surface, and then we rank those structures. In this part of what we're doing, we'll typically use what we call force field methods, where the interaction energy between atoms in our system is written in analytic mathematical form, so it's quick to evaluate. We can only really afford to spend seconds up to maybe minutes per crystal structure just because of the sheer number that we're dealing with. Okay. A key step then also is to identify and remove duplicate crystal structures. If we've done this initial structure search step well, then we will end up with duplicates, so many copies of the same crystal structure. We want to get rid of those so that then at the end we have a set of unique crystal structures. And I'll talk briefly later about the methods we currently use for identifying duplicates. What we end up with out of this process is something like this. So each point on this graph here, I'm showing its energy, And I'm showing here its volume, okay? So, and each of those points is a distinct crystal structure for that molecule. Okay? And if we're looking for the lowest energy ones, we're looking towards the bottom of that plot. So in that structure generation process, what are our degrees of freedom we're caring about? So what is the dimensionality of the lattice energy surface? Well, we have a crystal, so we have a unit cell. So we have the 3 lengths of that unit cell. We also have 3 angles, the angles between those lattice vectors. So that's 6 degrees of freedom already. Then we put a molecule in that box, and we can choose the position, another 3 degrees of freedom, and the orientation of that molecule. That gives us another 6 degrees of freedom per molecule that we put in that unit cell. If the molecule is flexible, then we may also have degrees of freedom that correspond to the geometry of the molecule itself. So we're talking about a search space which is typically, say, 12 degrees of freedom and upwards. And we're trying to find all of the local minima on the energy surface, which is the energy as a function of all of those degrees of freedom. So when we're using these sort of quasi-random sampling methods, we're trying to sample those degrees of freedom as uniformly as possible so that we can find the local minima across that whole surface in an unbiased way. We need to be able to quickly check for unphysical structures where we might have clashes between molecules to either reject or relieve those clashes. We use some methods that come from sort of computer graphics to sort of quickly spot clashes between molecules and maybe expand those out if there's a clash. And then any good structure will pass it to an energy minimizer and look for that nearest local minimum on the energy surface. So to have a look at how well this works, some people are surprised that we use methods that are essentially sort of a random structure approach, but they work pretty well, okay? So what I'm showing here are results for this molecule here in this particular space group, so the symmetry of the structures we're looking for. And here on the horizontal axis, we're looking at the number of structures, the number of energy minimizations we performed. And on the vertical axis, it's the number of unique structures. So we're screening out duplicates as we go during this search. Okay, and the different colored lines are just different variations on the method of how we transform our quasi-random vector into a physical trial crystal structure. So you're seeing there aren't great variations there, but after say a few thousand energy minimizations, then this plateaus, okay, which is telling us we're not generating anything new, so we found all of the local minima on our energy surface, or all of the potential crystal structures for that molecule. And that's where we can then probably safely stop that search. Another thing to notice is that we performed, say, a few thousand crystal structure minimizations. The number of unique structures we have is only hundreds. Okay, so we do need to be getting rid of those duplicates, but the number of duplicates that we have in there is also giving us confidence that we've performed a fairly thorough search of the energy surface. Another way of monitoring the progress of these searches is to monitor what is the current lowest energy structure that I have after a certain number of successful energy minimizations. And those are these dashed lines here. You can see actually that stops changing in this case after only a few tens of energy minimizations. And that's quite frequently the case, in that the global minimum, the most important structure on our landscape, is found quite early. And what we think this means is that global minima tend to have quite wide energy basins. We can also, if we're interested in not just the lowest energy structure, but also those that might be slightly above it, we can monitor, say, the mean energy of, say, the current 10 lowest energy structures. That takes a little bit longer to converge, but only if, say, a few hundred minimizations, and we have that, what looks like then a complete set of low energy structures for that space group and for that molecule. Okay, so these quasi-random methods actually work pretty well. We're currently implementing some things which bring this down a little bit faster, but we don't want to get rid of sort of the uniform and unbiased approach that we quite like during the structure search. So these are some plots to sort of show some things that we have to worry about when we do these searches. Here, for this molecule that we've been looking at, MACSMIN, what we've done is we'll oversample this deliberately just so we can gather better statistics. We do 10,000 energy minimizations. And now each of these sets of bars, again, the different colors are different variations on our method, shows the number of times we've hit each energy minimum. And the energy of the energy minima are shown on the horizontal axis. So these are the 10 lowest energy structures for that molecule. What you can see again is that the global minimum is found very frequently. Okay, but the frequency that we hit these structures varies structure to structure. So the next lowest energy structure, which is only just over half a kilojoule per mole above the global minimum, that's a very small amount of energy, is hit a lot less frequently. Okay, I show this second molecule here because this is where things can start to get worrying, because again, global minimum and now the second lowest energy structure are found very easily, okay, hit thousands of times within 10,000 energy minimizations. But the fourth lowest energy structure, which is within the range of structures we would expect to be able to see, perhaps from different crystallization conditions, is actually missed in some of these searches and is only hit very, very small numbers of times. Okay, so actually in terms of improving these methods for finding structures and looking for structures, we're not often looking at getting to the global minima faster, because we're already doing that quite well. We're looking at flattening out this type of distribution to make it easier to find these more difficult to find structures. Okay. Another way of looking at the same data sets, this is this problem molecule, quinacridone, that I was just showing you. That fourth lowest energy structure was infrequently found. Here, what I'm showing you is at each step in the Sobol sequence, which is the specific sequence we're using for that quasi-random search, showing you which structure we get to when we energy minimize this trial crystal structure we get from that quasi-random vector. And here you can see very clearly which are the difficult structures to find. And here I'm showing just two different variations on how we turn that quasi-random vector into a trial crystal structure. Here we had those problem structures. The number 4 was already from that last slide, looked hard to find. Also a couple other high energy structures. This one's found once out of those 10,000 structures. We slightly changed the way we do things in terms of that transformation from the vector to the crystal structure, and we can improve that in terms of making all of those low energy structures found in similar frequencies. So those are types of things that we try to do to our search. The particular change we made here was on the left plot. What we did was if we ended up with a trial crystal structure where we have overlapping molecules, we reject it, which seems the obvious easy thing to do. On the thing on the right-hand slide, what we do instead of rejecting is we adjust the structure just enough to relieve that overlap of molecules and then energy minimize. And what that tends to do is it actually lets us keep some of these trial structures which correspond to difficult or narrow energy minima on our surface. Okay. So simple things can end up improving things quite a bit. I realize I haven't shown actually any crystal structures up until now, really, so I thought I would show one, and this is just to show you what we want in terms of agreement. So here what we have in red is going back to this MACSMIN molecule, which was one of the first ones I was talking about, the lowest energy structure within that space group, and the second lowest energy structure overall, if we start to sample all common space groups, is the one that's shown in red. And what we get when we actually grow that crystal structure in the lab and get the structure from X-ray diffraction is the one in blue. And so you can see that the molecules are overlaying really quite nicely. Here, the RMSD in atomic positions is about 1/10 of an angstrom. So that's pretty good. And that's usually good enough for us in terms of going on to then predict properties. So I'll use that sort of as a segue into talking about comparing crystal structures, because we need this. I've talked about removing duplicates as we do our search. This overlay of this predicted and observed crystal structure shows you hints at one of the problems we have, is in that a crystal structure, how we represent a crystal structure is not unique. So the difference here that we see, the box, the red box and the blue box, are both unit cells for that crystal structure. The red corresponds to the predicted one, the blue to the observed one. They happen to just have a different origin set for them. But that would make it difficult if you wanted to just use, say, an RMSD of atomic positions to match duplicates or identify duplicates in a set. More commonly, you can get things, these two crystal structures here for a molecule that we're currently looking at. They don't look very similar at all if you look at those packings, okay? And here are the lattice dimensions. Okay, those are actually identical crystal structures. It's just that in what's on the bottom here is predicted crystal structure, and on top is the observed crystal structure. The definitions of the lattice are different. Okay, so the angles between lattice vectors and the lengths of the lattice vectors are chosen differently, but they represent the same crystal structure. So again, we couldn't just straightforwardly use lattice vectors as a way to identify duplicates. And we also have the symmetry which constrains our other angles to 90. Okay, we could have effectively an infinite number of possible lattices for a particular crystal structure. And as part of this also is that we have numerical noise in these crystal structures. So we're trying to identify duplicates when we energy minimized, say, 1 million crystal structures and brought them together. All of these have gone through, say, a Newton-Raphson energy minimization, where we're trying to find that local minimum. We stop the energy minimization when we hit tolerances, say, on the gradients, the forces on that structure. Okay, but if structures are coming from different directions on this energy surface, okay, they may stop with structures which are then fairly far enough apart that it makes a straightforward identification of duplicates tricky sometimes. Okay, because the gradients when you come from different directions on the energy surface can be quite different. Here I have a steep wall where this structure has come from. Okay, that might correspond to, say, atomic clashes between molecules, where the repulsion energy, which keeps molecules apart, goes exponentially with the distance between atoms or between molecules. Maybe I've had another structure which was come from a starting point where molecules were far apart and being drawn together by, say, dispersion interactions. Or here the energy goes as 1 over the distance to the power of 6. So it's much softer interactions. So you'll have shallower gradients. You'll end up stopping earlier if you just have a tolerance on gradients to identify when you're at the minimum. Okay, so then you have two structures. They've both finished energy minimization, but they're not quite at the same coordinates. Okay, so you need some tolerances when you start to try to identify these duplicates, because you want to take these and say actually they're the same structure, only keep one of them. Okay, so what do? We actually don't end up using the lattice vectors because of problems that I talked about a couple of slides ago. One method that we currently use is we take a molecule in a crystal, and because it's a crystal, then every molecule is in that identical environment. We build a cluster of neighboring molecules and we compare atom-atom contact distances. And we set some tolerance on that. And if those two structures, a reference and a comparison structure matched within those tolerances, then we accept that as a duplicate, they're a matching structure. The other thing we use, because it's actually quicker than this, looking at all atom-atom contacts, is we compare simulated diffraction patterns. Okay, so the diffraction pattern tells us about spacings between layers of electron density in our crystal structure, or effectively layers of atoms, and they can be treated as a fingerprint of our crystal structure. Okay, we have to, because again, we might have slight volume differences between structures, maybe due to energy minimization tolerances. Then we have to allow for sort of stretching of these powder diffraction patterns, and we use dynamic time warping to do that. Basically stretch these out to see if they would match if there was a slight stretch or compression of those spacings between lattice spacings. Okay, and the last thing I'll end up talking about is just a bit about these landscapes. I showed you a couple of examples of landscapes where we have all those dots corresponding to our unique crystal structures we get out of predictions. So a question you might have then is, these are a couple of simple molecules. Why am I showing that energy range? Okay, there are actually structures that go well above this, lots and lots of more possible crystal structures. And the reason is because we've looked at where we tend to find observed crystal structures on these energy surfaces. Okay, so what I've circled there are, for these two molecules, what are the observed crystal structures? Thiamine only has one known crystal structure, and it corresponds to what's pretty close to the global minimum. Theophylline has a bunch of polymorphs, and that's where we find them. So to inform this, we ask the question of how big an energy window do we need to look at. We've looked at lots and lots of molecules. Okay, so what we did was we pulled out known polymorphic pairs from the database of crystal structures. We pulled out over 1,000 crystal structures corresponding to over 466 pairs of polymorphs, and some with triplets or quadruplets. So we have 601 energy differences between observed crystal structures of organic molecules, just evaluated the energies and looked at what the distribution is, because then that tells us where we expect to find structures on our energy surfaces. And energy differences are small. Polymorphs, over half of known polymorphs are separated by less than 2 kilojoules per mole, which is tiny. Okay, and errors in the way we calculate energies can often be bigger than that, which is a problem for us as well. 95% of observed polymorphs are less than 7.2 kilojoules per mole. So that tells us where we need to sort of stop looking on our predicted energy surfaces. Once we're up at around 7 kilojoules per mole away from the global minimum, then we can probably consider the higher energy structures to at least be unlikely to be experimentally observable. So we can sort of turn that distribution on its side and treat it as telling us about the probabilities of finding structures as we go higher and higher on that landscape. I wanted to talk a little bit about dynamics, besides that actually crystal structures are not static. The molecules are not sitting in place, they're vibrating. I haven't left myself enough time, so I'll just show movies. These are vibrations of molecules in crystal structures. The energies of these vibrations are very small, okay? Thermal temperature, thermal energies at room temperature are about 207 wave numbers. These are all vibrations under 100 wave numbers. So they're very well populated at room temperature. So molecules are actually vibrating about, and that impacts on things like free energies and entropy differences between structures. But I won't be able to talk about that much. So hopefully that has given a useful overview of how we go about predicting crystal structures and a little bit about the types of methods we use. Some of the challenges we have now are I first did my first prediction, it took me about 4 months for a small molecule. We now aim to do these in a matter of hours, okay? And it takes quite a lot to try to get those timescales down. And it's not just big computers, but other things as well to think about. Matching duplicates, it seems like only a small thing, but it actually makes a big difference to be able to reliably remove those duplicate structures when we do a search. And interpretation of these energy landscapes is a big thing that we need to try to do, because these structures we get out, I'm showing energy versus volume plots. That doesn't tell you a whole lot actually about similarity or families of similar structures on our landscapes. These are just a couple of things I'm showing here, which is where we try to dimensionally reduce the sort of high dimensional set of structures and identify clusters or families of structures. And we're trying to work in those types of areas now. And I'll stop to try to leave a little time for questions if we have any. Okay.
Yes, thank you, Graeme. Let's clap for Graeme, please. Yes, thank you very much. So please, a quick question, 1-2 minutes. Any questions from Graeme? For Graeme?
I have a quick question, May?
Yes, please, Ron.
Yes. So Graeme, when you do the comparison by comparing the diffraction diagrams or the powder diffraction diagrams, how difficult is it to compare the actual diagrams and not to compare powder diffraction? And then if so, would it be possible to compare the Fourier transform, not the magnitude squared, but with the phases? Because that contains a lot of interesting information.
Yeah, so I guess the first part of the question was,
powder
could we just
versus
use
actual diffraction?
Yeah. We could, I guess it's a matter, it's probably a bit faster to do the powder because you're just doing a one-dimensional comparison of these patterns. If you're doing the full three-dimensional, you probably have to worry just about orienting to find the best match. Now, we worry about speed, and we find the powder pattern works pretty well. Second part, yeah. So I guess you could, and you'd have extra information in there.
This gauge function that I discussed in my talk is exactly related to how equivalent structures, I call them indistinguishable, how indistinguishable structures are related when you look at them from Fourier space. It's all about the phases that quantify that relation between them.
Yeah. So not something we've looked at. But
and then you might even have those homometric structures that have the same diffraction but are different.
Yes, and that we worry about, and I worry about using the diffraction.
Right. But you can expose them if you see the phases.
Yes, yeah. Yeah, so it could be, what we tend to use is the diffraction pattern comparison as a quick screen, then we check it with this real space overlay. But we can, I guess if we thought about what you're proposing there, that second check might be, because it would probably be a bit slower, but it would give us a bit more information. So it could be sort of a second screen, I guess, of do you happen, to check duplicates that we found. So.
Okay.
If I ever come for my sabbatical, then we can discuss that.
Okay, yeah, we should talk about it.
Great, great. So we are making connections. Very good.